Okay. Hello, good morning, everyone. So welcome for today's webinar where we are fortunate to have a true expert in the field of Western blot, Dr. John Yu, as our distinguished speaker for today. My name is Cassie and I'll be the host for today's webinar. Western blot is a fundamental technique in protein analysis, playing a pivotal role in the detection and analysis of proteins. It has been a cornerstone in countless research projects, enabling us to unlock the mysteries of the proteomic world. However, the multi-step workflow is complex and time-consuming. But today, we are fortunate to have Dr. John, who will give us a quick review of instrument-based one-stop solution to accelerate protein analysis through streamlining Western blotting process. The solutions can achieve publication-level quality Western blots results by conducting environment-friendly bench work in half a day. So whether you are a seasoned scientist or just starting in your journey in the lab, I'm confident that you will find valuable information and inspiration from today's presentation. So this webinar will be a recorded webinar and the recording will be sent to you after the session. If you have any questions during the webinar, you can type in the chat box and Dr. Yu will answer them after his presentation. So without further ado, we'll let Dr. Yu to start his presentation. Dr. Yu, the stage is yours.
Good morning, everyone. It's a pleasure to be here to present to you about Western blot and some of the work we have done on this field. Give me a second. It seems like it's frozen. Okay, so first a little bit about GenScript. And as you know, we are a biotech company and our motto is to make research easy. And we have a number of different business segments. And I am in the life science services and products division, where we product tools and services to research in life science laboratories as well as biotech companies. And as you know, we also have CDMO, cell therapy, and then also industrial products divisions as well along with us. So we're a global company with offices around the world, and then in Asia Pacific, we are based in China and with the office in Singapore. So on the product division, we have a number of product directions where all started from reagents, and one of the topic, the direction today is going to be the phage gels and protein analysis, which is the main topic today. And so it's actually been quite a journey from the establishment of GenScript in 2002. And along the years, we have been adding different products in this product divisions, directions, and also we're actually adding more into this product line to make Western blot truly easier for everybody. So first, what is Western blotting and why we need to do it? And a lot of times when we're doing a study of biological systems, so we're looking at certain changes and its impact in either the cells or tissues or a part of the animal. And then this is a lot of times represented by a specific protein. And so the measure of the specific protein on its expression and then on its levels are important. So to achieve this, there's multiple ways, but definitely Western blot is one of the cornerstone of this technique. It needs to do three things. First, it needs to separate the protein from the mixture by its size, and then it needs to get it immobilized on the surface so that it can be studied. And then we need to have a way to study it with specificity. And this is achieved by primary and secondary antibody. And so this technique has been in the field for a long time. And I know most of the Life Science Research Institute would have a lot of labs that's using it. So the current method, there's nothing wrong with the method itself. We goes from sample preparation to making the gel to separate by electrophoresis and then transfer it to a membrane, then, with all the antibody incubation and washes and all that step. And then when we finish that, we get the final product of a membrane with the signal that we're looking for. So if you have already experienced it, you probably have such feeling that, this process is actually really long. The whole process can take a full day, sometimes 2 days if you incubate overnight. And then in some of the steps, it can get messy. By saying messy, sometimes it's the setup. If you do the protein transfer with the wet transfer method, sometimes the lab can be messy because you need to make a lot of buffers, and then it has to be cooled in a bucket of ice, and that it melts everywhere, and that's one kind of messy. And another kind of messy is in the results. And here are some examples. And I take this from Rice University, where they have what they call a Hall of Shame for SDS page, where this is just everything that has been wrong with the SDS page gels. A lot of times this can happen because there's incomplete polymerization, because the density of the gel is wrong. It's just too dense. Because the loading gel was not poured correctly. It's uneven on the level. Use the wrong buffer for electrophoresis, and sometimes the buffer we used, if we made it ourselves and stored it for a long time, it can be contaminated, and that give it a pretty dark background. And the last one, sometimes if we load the samples with different salt concentration, on the same gels, we have some weird band shapes show up on the gels. This is a different kind of messiness that we could see in this, in this process. Now, how do we avoid that? A lot of times, we're trying to think about ways to do so. And for GenScript, as we're trying to make research easy, we are doing it in our own way. So first off, for the electrophoresis, let's assume we already have the samples. We need to get it done right and get it done fast. So, so first let's talk about this step. And of course, we start by making the sample first, and we need to have a sample buffer, and then, some of the other things that's associated with it, like the protein standards. So let's talk a little bit about sample preparation. And as you know, in SDS page, the separation is done by size. And this is two things need to happen. As protein, usually they're folded into a certain structure, and then it's actually a globular particles in the solution. And so we need to unfold this, structure by adding a certain detergent, usually SDS. That's the traditional way of doing it. And then we need to break down the disulfide bonds, which sometimes keep it, connected covalently. And so that's achieved by adding DTT or BME, some type of reducing agent. And therefore, we can have a linear peptide-shaped protein where we can separate. So the detergent used here actually makes a pretty big difference, and I'll show you in a little bit better. And so laminate buffer is the one that typically uses SDS. Now, a new recipe would use the LDS buffer, and this recipe will be available in GenScript website. You can look it up, and so LDS is a different detergent, which the structure is slightly different, which is a little bit longer than the SDS, and they give a much better linearization of the protein, and the results is a better resolution in certain parts. And this is a comparison of two lysate that's using a different buffer to prepare. On the left is the laminate buffer, which is using SDS. On the right is LDS buffer, which uses LDS, and everything else is similar. And if you're looking at the gels, you see just on general, the resolution is better with LDS. And on the right hand, if we zoom in, as you can see, you'll have a slightly better resolution on the LDS buffer side. So this is one of the first recommendations, is that if it's possible, you can use LDS buffer for your sample preparation. If it's available, you can either purchase it or can you make your own, which is fairly easy. Now, comes to the gels, and I know still majority of the labs still making the homemade gels with their own reagents and with their own devices, and it doesn't take too much work, but then things can always go wrong. That's because the solutions sometimes has a expiration date, because the polymerization reagent has it's more sensitive to oxidation, because buffers can have things grow in it, and that all causes problems. So one of the solution is to have it pre-made. But to pre-made a gel is not enough, because it doesn't make it faster. To make it faster, we need to do a few things. So the first thing we did is to use the Bistris buffer system instead of the traditional tris-glycine buffer system. So there's a few facts. I don't know if you already know it. And the conclusion is that Bistris buffer system will give you better stability for your gel. And why is that? Okay, that's because in SDS page gels, it's formed by polyacrylate polymers, where there's a lot of CO2 amine bonds. Okay, these bonds is suspect to hydrolyzation under alkaline solutions. So when we make a traditional homemade gels, we use the tris-glycine buffer, and the separation gel pH usually is around 8.3, which is alkaline. This means this hydrolyzation, once you make the gel, will continue to happen, okay? And that's why, it's not recommended to use gels that's more than a couple weeks old, because, things can happen in the gel that you don't even notice, and the gel will swell, and then the separation will, be affected. So, if you stretch it, you can use tris-glycine homemade gels by about a month. But then if you switch to a neutral buffer system, which is like Bistris, and you can use it for much longer. And in our practice, we can actually store gel up to 18 months from the date of production. But then, typically we can store it over a year. So to make it run faster also is the requirement to reduce the experiment down to 3 hours. And we do that by increasing the voltage where we run the gels at. Typically, we can't run the gel that fast, because there will be a lot of heat generated, because that generated heat will affect separation and then make the gel look weird, and then your result would not be trustworthy. And by tweaking the gel content and then the buffer system, we were able to produce a gel that's stable, can be run stably at 200 volts. And that means you can finish a gel roughly around 20 to 30 minutes, and depending on the buffer that you use. And we use two different kind of buffer system, the MOPS and MES based buffer. And there's a difference, which I'll come to a bit later. So, and another special feature about the gels that GenScript offers, and of course, you can use a typical homemade gels with two slabs of either plastic or glass, where you make the gels, and then you can load the sample in between the two plastic or glass. But that limits the amount of sample you can load into this well. The GenScript gel has a special design where we enlarge the loading well by making the plastic enlarged at the top. And this gives you a much larger loading volume compared to a typical gels. And this is especially useful when your sample is diluted, where you have to load a large volume. Okay, so that's the third thing unique about the SurePage precast mini gels. And so, of course, we offer a range of different percentages and then also the gradient gels. And I think for most labs, a gradient gel would be a good tool to have, where it can cover a large range of molecular weight, and regardless of your sample size, you'll still be able to separate out the gel. And we have different well format, so we have the two buffer system, and then they can run roughly for MES at 20 minutes and then MOPS at 30 minutes. And this is when you run it at the top speed. And then the loading volume, we can get it up to 80 microliters, which is good for diluted concentration. Okay, now how to choose between the two buffer systems, as we mentioned. The two buffer system actually makes a difference when you use with the SurePage gels, the precast gels. The gels will separate better for large protein sizes when used the MOPS buffer, and separate better for smaller sizes proteins for MES buffer. So here is a gel that's breaking into two sections. The HMW is the high molecular weight size part, where it's the top of the gel, and the LMW is the low molecular weight. Okay, and this is more of a illustration. It's not exact, but then it shows the point. So on the MOPS buffer side, you'll have a much better resolution for the same band at the higher molecular weight range. And on the MES side of the separation, you'll see the lower molecular weight would have a better separation and the resolution. Well, for Western blot, this typically is not a deal breaker, because you'll have a specific way to detect it with antibodies. So you can use both, but then always good to have better resolution, just in case there's something that's interfering with the antibody that give you a false signal, right? So, okay, so when choose the gels and the buffer systems, and this is something you can keep in mind. And although in most cases for Western blot, both would work just fine. Okay, and one thing I have to mention, and if you're using trisglycine homemade gels now, and you decide to give the precast gels a try, do not use your trisglycine running buffer, okay? Because that will not work. Okay, the experiment will give you a very strange results, because the two running buffer from the two buffer system are not compatible. So use the buffer that we sent you, either the MOPS or the MES, and they'll give you a better results. Okay. If you do want to switch over and give it a try, and if you already have Bio-Rad tanks, and good news is that you do not have to get another tank. And you can just simply flip the gasket, and then you're set to go. And so all these tanks can be compatible. If you're using other tanks right now, you can get the Gymbox tank to give it a try. And we do have right now a promotion. I think with the order up to 10 boxes of gels, we can give a free tank, and that's a promotion that we have been running for a long time. And so take the advantage of it if you do decide to give it a try. Some of the examples of the SurePage precast gels and the results. And here are some of the comparisons. On the left-hand side is the result for you know, a Kumasi stain. And as you can see, the consistency is fairly good, and you know, across the board with just both the lysate and also the protein markers. On the right hand side, this is the Western blot results, where you know, we have the Western markers on there, and then we have the signal down there in the zero dilution, and it comes up quite nicely. And we also have already published a good number of high impact papers in you know, different type of journals, and this year we also received a lot of new publications coming back to us, and we're still compiling that, and we can put that up in our website and to share it with you. Some news also, and as you know, we were just talking about mini-sized gels, and sometimes that's you know, good enough. Sometimes that's not that good, especially when you have a lot of samples to run. And so we're developing a new precast midi-sized gels, and which typically come from either Bio-Rad or thermal, but then we'll start offer that in sometime next year. And then it'll come in 20 wells and 26 well format, and it'll be compatible with the you know, the existing tanks that you already have. So just a pre-announcement, and we'll give you more details when it's launched. Now, complete with the gels, now we have reduced electrophoresis from a few hours, let's say, you know, 1 hour to make the gel and an hour to run the gel, to roughly about 10 minutes to set up, and then half an hour to run the gel. So that's about 40 minutes. And then we goes to the transfer. And we need to do that faster too with a device. So we have this device we call blot L1 protein transfer system, and which can finish a transfer in roughly 9 to 17 minutes. And a typical experiment finishes around 10 to 12 minutes. So before we get into the device, I think we need to talk about the principle of transferring proteins. There are two ways to transfer the proteins. The first is the what we call the traditional wet transfer, where a tank will be filled with buffer, and then it'll be immersed with ice to cool it down. And you know, the gels, the membrane, and then the pads will all be packed together into this tank with and then voltage will be applied to it, and the protein will be transferred. This is the standard way of doing protein transfer, and I think a lot of you might already had experience with it. And this is sometimes, I feel, you know, can be a little messy, especially with all the buffers and all the ice and melting everywhere. But it's a good method, and it's the, you know, the results is still considered as gold standards, and then the stability is good, and it covers all the protein ranges, and it's a bit slow. So sometimes it takes, you know, an hour and a half. Sometimes it takes overnight, depending on what experiment you're running. So based on the wet transfer, there's another, you know, group of devices that runs what we call semi-dry. The semi-dry can increase the speed significantly, reduce the transfer to roughly 10 to 15 minutes. But then because the semi-dry transfer reduced the amount of buffer, and then you know, couldn't dissipate the heat generated, and so it has oftentimes had trouble to transfer the large molecular weight proteins. And so if your target is you know, small, and I think semi-dry would work just okay, and it's convenient, so everything comes in pre-packed you know, kits, and you can set it up pretty easily, and you know, the stability is okay, but for large molecular weight proteins, you might run into some trouble you know, for transfer efficiency. And so when we were developing this product, we think, well, why not to, you know, have, you know, the best side of both worlds, right? And we want it to be fast, and we want it to be able to be compactable compatible to all sizes of proteins. So that's why we have the eBlot L1, what we call fast wet transfer system, where we actually do not limit the amount of buffer we use. We use more buffer than the semi-dry, and that's a fact. But then because of that, we can be compatible to all sizes of proteins, that we can transfer proteins very efficiently, even if its size is large. And we can do it fast, in about 10 minutes. And then the stability is good between different runs. Okay, and so I think the features, and I won't read through it. I think the one thing you do need to remember is this is based on the wet transfer principle, and it's the only one that's currently on the market that offers the wet transfer based fast transfer device. Okay, and then we have a couple of videos online, and once we have this slide shared with you, and you'll be able to go online and find it, and you can watch it. And we have a promotional video, and then we'll have a tutorial video that explains the details in terms of how it works. Some of the results that we get with comparison from different devices, and then the left side is the wet transfer, and then the right side is the eBlot L1, and then in the middle is a couple of semi-dry transfer method. As you can see, on a lot of this device, especially on even smaller transfer targets, eBlot is doing if not better, but as well as the wet transfer. And when you come to the larger size, and eBlot is doing just as good as the wet transfer, the standards. But you see in the semi-dry, sometimes it does a little bit, less of a perfect job. So, and this is an example we have been talking for quite a bit, if you have heard of it, and I will just repeat it quickly. And this is a researcher in United States where he has been studying very large protein complexes, and he's been trying all kinds of devices on the market, and he's trying to transfer a protein complex of about 1,200 kD, which is extreme, okay? And not every lab would run into such a target. And when we launched eBlot L1, he found us, and then he wanted to try it. And so this actually is not done with a standard protocol. We actually did make some adjustment to to make it work better for large protein complexes. And then so with this, and he, in his words, he got the best results and he ever got with this complex, even better than the wet transfer that he used. So well, this just demonstrates that we can transfer large proteins very well. And also if you happen to working on a complex on similar range, and just get in touch with us, we can help you to get it transferred. Now, after we transfer the protein, which we reduced from, say a couple hours to about 10 to 20 minutes, now we need to do the incubation. And this is a tedious process where, different reagents have to be added, and then incubated, and then washed a few times, and then on the shaker. And so this is doesn't feel like a very productive process. So we wonder, well, why we can't let a machine to do that, right? We don't have to do it on our own, and with the timer and sit around, not be able to walk away. So we developed the Easy West automated West device, and this is a device that will do the incubation in your place. And so this shows the typical incubation process, where you add the buffer, and then you walk away a few minutes later, and you come back when the timer beeps, and switch out the buffer, and then come back again a few minutes later. And instead of doing that, we can use Easy West Lite to do this for you. And then it's an automated system, and it can do, take the membrane, incubate primary, secondary antibody, and then get ready for the detection step. And then we made it into an open platform where we made it compatible to whatever buffer system you're using currently, so that you do not have to switch to a proprietary reagent. And right now, this device only can process 2 membranes at a time, and it's compatible to mini gel size. And the amount of buffer or the antibodies volume used is about 10 milliliters. And so it's a pretty compact system, and it'd be easy to find a spot on your bench for it. So the way it works is that, set up the solutions, primary, secondary, and blocking solutions in the, in the machine. Take the cassette where the membrane goes in, and with a couple of spacers to hold it in place. And once you close it, insert it into the device, and then set up the program and click start, and then you can just walk away. And when you come back, you'll see some of the results. And these are mostly, the results we got in our lab, and compared in the conventional method with the results from EasyWest Lite. And as you can see, they're pretty comparable. So another product that's coming along with the EasyWest is what we call the EasyWest diluent kit. This is a kit that you do not have to use, but then you can to use it to shorten the antibody incubation time. So this kit comes in a solution, and this solution can be used to acting as a blocking buffer. That's the first thing. But then the primary reason to use it is to use it to dilute your antibody into it. And so with this, you can shorten the antibody, incubation time by about half. And so I think a third to half is what we typically goes for. And because antibodies are such a variable reagent, and we cannot say for sure all antibodies have been tested and work on it. But we have tried a lot of antibodies, and they all work just fine with this reagent. And so yours should be, work with it just fine too. And so the reason to use it is twofold. One, the blocking time can be shorter. And then the second thing is you can short the antibody incubation time, which is the longest time that you can have on the whole antibody, process, on the Western blotting process. And, sometimes this takes 4 hours or to 5 hours, and sometimes this take overnight. And that makes it a 2-day process. And so it'll give you a good and consistent results with the, even background. And just show you the results, and we compared the, side by side, the, traditional way of doing it with milk, and, with the standard protocol where we incubate primary for an hour, secondary for an hour, and then, add everything together, including the shaking, and that's about 4 hours. And then with the diluent kit, we skip the blocking solution, because this will act in as the blocking solution in the protocol. And then we have the primary antibody time, and then we have the secondary antibody time. And then in the end, we spend roughly about 2 hours to the incubation. Now, here's the results. We have tested beta-actin, tubulin, HSP70, beta, so these are pretty standard control markers in Western blot. So they all seem to work just fine with the diluent kit. Okay. So the last thing that is not necessarily directly connected to Western blotting is a staining device where it's under similar principle as e-blot, but then it's for staining the gel. Because not all gels are done for anybody. Sometimes we just want to see if our purification are done correctly and we got our protein and how pure it is. So of course, we have the traditional way of doing the staining and then, sometimes microwave to make it faster. But then the smell and then also, the shaking can give you some headaches. So this device can make it happen in a shorter time within 10 minutes, and then it's fully automated, and it's based on the Kumasi stain. So the gel will come out similar to what you get with the Kumasi stain. So the sensitivity is roughly about 12.5 nanograms. Okay, so that's a decent sensitivity. So some of you might have already tried some of the staining buffers on the market, and then some of them claim to be fast, and some of them are. But then the sensitivity they claim is under the best circumstances, and oftentimes if you use it in the fast protocol, and the sensitivity doesn't nearly goes to what they claim the sensitivities are. And also another thing is about the consistencies. With the staining buffer, we have tested in the house as well. The staining buffer doesn't give nearly as consistent results compared to the the device because it's not as controlled. A lot of factors like temperature, like the amount of buffer, like the shaking speed, like the time, shaked, and is not really controlled that well, and that affects the consistency. So that's one of the other advantage of the device. Okay, so it'll give you a good and sensitive results, and also in a consistent way. So compared to a manual staining, and of course, there's no comparison, especially in time, we can get this done in roughly 10 minutes, while the typical stain will give you, 3 to 6 hours. Even with a fast stain, it'll probably still be faster, longer than that, than 10 minutes. And here are a couple of results for the staining results with the e-stain device. And on the left, this is taken from an imager in just the black and white type. So, there's no color to it. And it's a zero dilution. As you can see, it can get all the way down to 12.5 nanograms as its limit to sensitivity. And then on the right-hand side, you have a comparison of a gel running same kind of samples across the board. And the consistency, across the board is very apparent. Okay, in addition, we, this doesn't come with a product. It's more of a protocol that we offer. This is a pencil stain for looking at the membrane to see whether it's, successful in protein transfer or not. We developed this in-house, and we gave it, published it on our website. To be used with our device. And so we do not offer the kit, but you can use it for free. Okay, so with that, I think we come to a summary where, if we look at a typical Western blot, process, each of the step will take a few hours. And, add them all together, that limits you to get 1 blot a day at the best, and which is sometimes, a limiting factor, it takes longer for you to get your results. And then sometimes it's just boring, especially like the incubation or washing step, and you're not doing, spending time more effectively. So what we did is to try our best to use various ways to shorten time, and to use the pre-casted gels, shorten the electrophoresis time by up the voltage of electrophoresis. And then we made the device to help transfer the protein under the wet transfer principle at the same efficiency, and then with much shorter time, which, roughly around, 10 minutes. And then we automated the process to incubate the primary and secondary antibody. And then with the combination of what we call the rapid kit, we could also cut down the time of incubation. So all in all, well, I have this in the wrong way. So we can get we can get the Western done in less than 3 hours. And so add all this together, and if we say this is a half an hour, this is 20 minutes, and this is 2 hours, and that's roughly 3 hours. So it's within 3 hours that we can get to the imager, we can get our Western blot results. And then don't forget that sometimes we can also make our gel staining faster and more consistent as well. With that, I thank you for your attention and then for your time. I'd be happy to answer any questions that you might have. And I see there are a few in the chat box. Let me have a look.
Yes, Dr. John. Yeah, thanks for the presentation. We have a few questions over here, so maybe we'll go through it one by one. The first one is from Kia. Is there any protocol for running EasyWest diluent kit in EasyWest automated Western device? What's the difference in comparison with traditional reagents?
Well, yes, the short answer is yes. So EasyWest device is compatible with your current protocol, regardless what type of blocking, what type of antibody, and that you're using right now, and it can be adopted to those solutions, and it should work just fine.
All right.
Compared to traditional. Oh, okay. No, well, so it's compatible with the traditional reagents, but also if you are looking to get results faster, you could also keep the rapid kit around just in case that it's needed. So, okay. Second.
All right. Thanks, Dr. John. The second one, is it possible to use diluent buffer during the manual Western blot?
I think so. It's the same principle. It's just that with the device, it's more automated and it's more consistent. With the manual method, it's going to be a little bit more variable, but it's something definitely can be tried, and I think it should work just fine. Yeah.
All right, thanks, John. The third question, for EasyWest, is it possible to hybridize different antibodies at the same time and using the cut membrane, not the standard membrane size?
I see. So the answer to this question is twofold. One, you can use, the cut membrane is fine. You can use cut membrane in the device. Now, because of the limitation of how many buffers you can include in there, it only works with one set of primary and secondary antibodies. So, if there are different sets of antibodies, then you're gonna have to run it consecutively or on a different device. So that's the answer to that.
All right, thanks, John. The next question is quite specific. Would you be able to share how much are the equipments and also the precast gel and also the buffers?
Oh, for that, I would say get connected with our sales representatives. I think they're probably more knows more details about the pricing and everything. I can just say we're competitive in all aspects, and so okay, and I think I'll keep it at that. Yeah.
Can. Yeah, Nabila, I will connect you with our local sales and probably we can give you like a price quotation for that. So let's move on to the next question. From Fatma, she have a question about the device that is used to block the membrane. How much blocking solution or antibody need to cover the membrane, and is it available for any types of membrane?
So typically for Western blot, it uses either PVDF or nitrocellulose membranes, and both can be used with Easy West. Now, in terms of the amount of buffer, it'll take roughly about 10 milliliters for primary and secondary antibody, and as well as the blocking buffer. For rapid kit, you don't have to use the blocking buffer, so that's skipped. Yeah.
All right, thanks, John. The next question from Nabila. With your automated blocking device, can it detect two antibodies at the same time? For example, your antibody of interest and control, like GADBH or tubulin?
Right. I think that's the same question as the one before, where so the answer to that is that we can currently just do one set of antibodies on a device. Now, I understand that typically we do experiment. We have our target, protein, and we also have internal controls. We actually do need to run, two set of incubations, and right now with this device, you're gonna have to run it, sequence or on different devices. But then, in the future, we're looking into developing a device that can do both at the same time. But then it's not, yet ready. So.
Okay, thanks, John. Next question from Ranjana. Do manually made gels, is it compatible with eBlot?
Manually made gels, I would say for the most part, it's compatible. However, because manual made gels are not, controlled, in each step as the precast gels, and sometimes you'll run into issues. We had cases where, a lab tried manual homemade gels on eBlot. And a lot of time it worked, but then occasionally they'll have gels that's kind of melted a little bit on the device. That's mostly because of the gel that, the process that making the gel is not as well controlled. So occasionally, you can run into a small issue with the eBlot, but you can use it and it's compatible. And we have labs that's doing that as we speak it. So.
Okay, thanks, John. Next question from Yung. Is the EasyWare software GelDoc, is it sophisticated like the GelDoc software, like BioRec GelDoc? Let's come.
I would say I think they're a little different. I think we do have enough flexibility to be able to get compatible with all the device, all the protocols that you typically run. But I think they're different in nature. So.
Okay, Ken. Thanks, John.
Yeah.
Next question from Jantakan. Does the EasyWest have a shaking system for membrane incubation?
Right. And shaking is what we typically do for membrane incubation. Now with EasyWest, we're using a buffer circulation system. That's usually, with a pump. That's not with the shaking, but it does the same thing, that mixing the solutions and then get in contact with the membrane and keep it incubated, and to have efficient interactions between the target proteins and then the antibodies. So yeah, not shaking, but it does a good job just as well.
All right, thanks, John. Next question, is the Easy Wax compatible with nylon membrane?
Nylon membrane. I'm not sure nylon membrane. We haven't not tested, but we can I can make a inquiry and see if there's information on that and get back to you. Okay.
Okay, yeah, we'll get back to you again. So next question, can we run and transfer the gel manually and then use Easy Wax?
Sure. Yeah. So I think each of the steps I just mentioned can be looked at independently. Then, you can adopt the, precast gel first, or you can adopt the blot first, depending on your situation. And they are, most times, compatible to the current, workflow that you're using, and they just replace a part of it where it can be connected into any type of current protocols you're doing, so long as it's roughly a standard Western blotting process, and they should be able to take part in there. So yes, it can. Yeah.
All right, thanks, John. We are down to our one last question from the audience. So can we just go ahead with the Western blot, the e-blot system, since we already have the BioRep Proteome system with us?
Okay, yeah. I think this is similar to the previous question where, whether one device can be adopted individually. I think, yes, that's definitely, that's can do. Yeah.
All right, thanks, John. Yeah, so we have already answered all the questions from the audience, but let's say if anyone still have any questions for Dr. John, feel free to shoot us an email and we'll answer them via email offline. So as we come to the conclusion for this webinar, I would like to express my heartfelt gratitude to each and every one of you who joined us today. It has been a truly insightful and engaging experience, and I hope you have gained valuable knowledge and inspiration from our distinguished speaker. I also wanted to extend our deepest appreciation to Dr. Yu for your outstanding presentation, and we are grateful for your generosity in sharing your knowledge today.
Thank you.
So,
Well, just one last point. I think
sure.
I think I forgot to mention, we do have sample packs of short page gels, the precast gels for for testing. And if you do have currently using BioRad gel tanks, and reach out to us and we can send you a free pack and give it a try.
Yes, correct. Send us an email if you would like to have a precast gel sample, and we'll ship it over to you to try it out. Yeah, so for today's webinar, right, it's a recorded webinar, and the recording will be made available to all the registered participants in the coming days, and we will also share the slides from Dr. Yu to you. So this will be an excellent source for you to revisit the content, and also you can share it with your colleagues who couldn't attend the session today. Your feedback is invaluable to us as well, and it will help us to improve and shape our future events. So shortly after this webinar, all of you will receive a survey form, and I kindly request you to take a few moments to provide your thoughts and suggestions for us. Your input will guide us in delivering more informative and engaging webinars in the future. So once again, thank you everyone for joining this webinar, and we'll see you again in our next webinar. Thank you and goodbye, everyone.
Thank you again. Bye.
Thanks, John. Bye-bye.